thank you to the speakers and thank you to participants to come here to actually the second workshop, Instantiation of Computing Among Cells. And so this is organized by Manish, who you can see here, and by Tumors, who's waving the cameras there. And our goal is actually to bring together people that are interested in distributed computing perspectives on biology, whether they come from computer science, biology, or in between, or whatever. And we are very happy to have Jeff Hasty as a first speaker. And of course, there's no reason to introduce him to the biology people. I guess all of you know him, but perhaps quickly for computer science. So let me just check. So he has actually a training as a physicist from Georgia Institute of Technology, if this is right. Then he did a postdoc in Boston University, and now he's professor at the University of California, San Diego, and director of the Biocircuit Institute and co-director of the UC San Diego QBio specialization program. And he has authored seminal papers on pioneering works in synthetic biologies, and we are excited to hear some of this soon. Thank you.
Great, thanks for the introduction. And let me just say very genuinely, this is the type of crowd that I like to talk with, because I can share some of the modeling and I can get into some of the nitty-gritty of how we go about our business with the quantitative methods. And I hope you see a flavor of this. A general theme I think that I would like to hit is, and you'll see this, I hope, emerge by the end, is the importance of basic science. So I organize a quantitative biology meeting. I've been sort of very much trying to move biology in more of the quantitative direction. It's not an easy lift because people often ask, so what are the examples of quantitative biology paying off? You know, so when you're trying to get this funded, or you're trying to get people to come to the table to support this, you know, you're usually talking to, of course, you're trying to grub around for money. And so, you know, at the end of the day, they want hardcore examples of where basic science turned in. You know, so now we have a really, really good one with CRISPR, right, turning into the Nobel Prize. That is just a wonderful example. But to a much lesser degree, I hope that when you back off and look at my talk, you'll appreciate that basically for 10, 15 years of my career, we were playing around with genetic circuits, trying to figure out how they work, trying to build oscillators with just a curiosity. And, you know, the NIH was very, very generous in supporting this for many years. And then the application fell into place. And now I think you'll see where we're going. And I hope that sort of that comes out as sort of how this started and the importance. I know I'm preaching to the choir, but so just to sort of put a pin in that, you know, we're known for building genetic oscillators, for engineering them and building them. This is kind of the laundry list of why in a kind of somewhat chronological order. You know, as a postdoc, I was lucky to land in Jim Collins's lab, you know, right when he was saying, hey, there's this thing where now we can engineer possibly gene circuits, you know, and I was just so lucky to land in that position as a theoretical physicist, knowing nothing about biology. And I took to kind of how do we model these things quickly. And because of my background in nonlinear statistical physics, I was particularly interested in oscillators and modeling oscillators and trying to build them, and then understand the functionality, the components. Synchronizing populations became the next thing. I'll talk about all of these things in some small detail, and then we'll get to the application, which is the producing and delivering of bacterial therapies. And then what we've done since then is to work on small bacterial ecologies in the context of these therapeutic delivery mechanism. Okay, so, and oddly enough, I end up sort of working in solid tumors, which, you know, if you told me that when I was a theoretical physicist, I would have said you were crazy. There's no, this is an alternative universe. This is not where I would end up. But oddly enough, that's where basic science can take you, and you just never know. Just to highlight this point, these are from my notes from 2000, like 1999, 2000, right? So I was thinking about positive feedback and bistability back then. Here was just the basic thing. Again, I like talking to this crowd because you kind of can get it. You know, positive feedback, you just model it in sort of the most basic way, and you can generate bistability as a function of some control parameter. So bistability can be used as a switch, can be used for a threshold mechanism. And in the context of oscillators, I was interested in adding negative feedback at the time in order to generate sustained oscillations like a hysteresis oscillator. And from my background, I knew this well as a relaxation oscillator in the context of neuroscience. So this is how neurons essentially work. It's a slow, fast system. Fitsu-Nagumo, those kinds of equations are just basically this. If you sort of scale back a little, mod the details, it's basically the same kind of behavior. So this is where I started as a postdoc with Jim, very interested in that kind of oscillator. Trying to build this with genes. So I started my gig at UCSD and we built one of them. So this coupled positive and negative feedback oscillates very nicely. And I apologize if you see my talks and if this is too much of a trip down memory lane, but I think it's important to get to where we got just by a little bit of highlights of the older stuff. So this appeared to work great, but if you look very closely at single cells, it sure doesn't look like that. It's pretty noisy. People have been talking a long time about, okay, cells, people make the sort of, I would say, bold statement that cells are noisy. I think they are noisy if you're looking at certain aspects of cells, but it may be that we're not looking at the right thing and they're not. I don't want to get into that philosophical debate about whether they're truly noisy or not, but at least they appear noisy to us when we build things. And so for, you know, in the early 2000s, mid 2000s, late 2000s, people were thinking about how do we circumvent this noise? You know, how do we intracellularly build feedback and do things to get around the noise? And, you know, it just looked too hard to me because you're trying to circumvent the noise with noisy things themselves. And so I'm sure people have made progress on that. I haven't kept up as well as I probably should. I'm not particularly great at literature. Anyway, the thing I was thinking, well, let's go straight to coupling cells together to get deterministic behavior rather than trying to get single cells to be less noisy. That seemed easier to me. So generate coordinated behavior at macroscopic levels at the colony level, as opposed to trying in single cells to get things to work. And so we had also thought about this in 2002 with Dave McMillan, who was another postdoc at the time. We had a lot of fun playing with quorum sensing, figuring out how we could take these oscillators and use quorum sensing to get them to synchronize across cells. And, you know, you just never know, but nearly 20 years later, who would have thought that bacterial quorum sensing would be my go-to circuitry? So, you know, I really am the kind of scientist who just keeps plugging away at things till I understand them really well. I don't have a lot of breadth in terms of like, oh, I didn't know he was working on that, or, you know, there's continuity. I just keep kicking at things. Some of this next bit will be sort of our voyage through quorum sensing. So let me just tell you what quorum sensing is for those who are not from biology. Basically, cells can coordinate their behavior via quorum sensing, what we would think of as synchronization. At low densities, cells are thought to have more individualistic behavior, but then at higher cell densities, they begin to talk to each other and coordinate their behavior. And I'm, of course, not the person to be talking about quorum sensing as a biological phenomenon. Bonnie Bassler would be the expert at Princeton. If you wanted to look up some really cool seminal papers on quorum sensing, Bonnie would be the person to look at least from my perspective, and maybe as a first look. For us as engineers, computer science physicists, whatever we are, the quorum sensing has some attractive properties. So the key ingredient is that there's this small molecule AHL that diffuses in and out of the cell. So cells, just think of each cell as producing some AHL. Just think of it just as constitutive, just constantly producing it first, and then we'll add some network here in a minute. And so the AHL goes in and out of each cell. And so if you looked in the media, the amount of AHL that would be there would be proportional to the number of cells. So you can think of it this way: the cell density, or the number of cells, triggers the amount of AHL, but then this AHL effectively can turn on genes that then trigger quorum sensing molecules. So the small molecules activates transcriptional regulation. So it's taking a population level information and transducing it to the genetic level. So population to intracellular gene networks. And it does it in an interesting way, and this is why it's called quorum sensing. So, you know, of course, we kind of have an intuitive understanding what quorum, the word quorum means. Those of us who are faculty members know that we can't seem to do anything unless we have a quorum. And even when we do have a quorum, I guess a lot of times we have trouble getting anything done. But the reason the quorum sensing, the quorum behavior occurs, so is that as cells grow, say exponentially, I've drawn cells growing, and this is more just a sketch of an observation, right? Is that if they grow exponentially, the quorum sensing molecule turns on more threshold-like. So so it's a sensing system that's converting an analog signal to a digital signal. And that's more in our language as opposed to, you know, how a biologist would explain this. But that's okay. Let's see how far we can see how useful that is to us. So how is it doing that first? You know, because there's some magic under here, right? Because if it's just constitutive expression, you're just going to get correlated behavior between N and Q. So there must be something going on, right? And it's back to positive feedback. So most quorum sensing systems use positive feedback to generate their switch-like behavior. So how does that work then? If I generated a phase diagram, and I'll write down some equations, I promise, but let's just look at Q versus N, output versus input, you would get the typical S curve in terms of a bifurcation diagram. And you can see how this would possibly work then. As cells grow, they move slowly on this, what we would call a manifold in nonlinear dynamics, right? And then when they hit the bifurcation point, that's when the quorum threshold is triggered. So the system goes on. So this is a bistable system. It's just a traditional S curve built from positive feedback. That's what quorum sensing is using to transduce this analog to digital response. So what do we do with this? In my lab experimentally, we synchronize. So this is just to put some meat on the bones here in terms of the actual circuitry. We use positive feedback from Vibrio fischeri. It's a marine bug, which has the quorum sensing AHL is the small molecule we were talking about. We put this into E. coli. And we built a coupled positive and negative feedback system where AIIA is a protease for the AHL. And because the AHL diffuses in and out, each cell is coupled to every other cell. So you have synchronization across the population. So you can see how this could lead to synchronized oscillations if the core circuitry oscillates, and it does. So this is what it looked like in microfluidic chips. This is just all the cells oscillating in synchrony due to the quorum sensing. And you can see the original motivation was to try to get deterministic like behavior, something that was easier to get my hands around in terms of engineering. as opposed to what we were typically seeing at the intracellular level, even though we saw kind of nice punctuated cycling. At longer length scales, you see this sort of wave-like behavior. These movies are, I think, on the internet, various places. And they're interesting. And, I think probably we didn't follow up and have some papers after this and study it more carefully. We just kind of moved on, and I'll show you why we moved on. But, if you wanted to synchronize things over a longer distance, this is kind of a problematic mechanism for that. Right. So then we took the quorum sensing a bit further. So this is the original quorum sensing system at the colony level, where the mechanism's too slow for anything beyond, say, you can even see some waves coming from top to bottom in this colony in the microfluidic chip. So let's just keep it in microfluidic chips because it's easier to think about. So just think about 4 colonies here doing their synchronization at the colony level, but not synchronized across colonies. This would be typically what we would see in our microfluidic chips. Would be, say, 4 of them, and they're not talking to each other. Okay, and quorum sensing can't work over those length scales, even if we had an open chamber. And there's some connection here due to the media. So quorum sensing molecule too slow to couple those colonies. So each colony is governed by the same mechanism that I discussed before. That is media coupling through the AHL. So what we found was there's a vapor signal that can be used to turn on the quorum sensing machinery, so that each time the quorum sensing colony fires, it releases a large pulse of the vapor signal, which then couples the colonies together. So it's synergistic coupling because you couldn't just use the vapor signal because it's too weak, it would diffuse too fast. You need the quorum sensing to get the colonies doing their thing to generate enough vapor to do their thing over the larger length scale. So this is essentially how it works. Each colony is kind of doing its own thing, and then you generate enough H2O2 vapor to synchronize the colonies together. So we played with this, had a lot of fun. We call this biopixels. And over centimeter length scales, we could generate what began to look like synchronized colonies. Now, there's some plumbing problems, microfluidics, and again, we didn't probably follow this up as much. We have a project going on right now that's related to the more recent circuitry I'll tell you about. But anyway, this is what the quorum sensing sort of just basic science opened the door. We use this for a biosensor at macroscopic levels, done a lot of stuff with this thing. So now let me get sort of closer to the present, the synchronized lysis circuit. So the original motivation here was to release a product and maintain cells in an optimal growth state. It was kind of broadly motivated. We definitely weren't thinking about cancer at the time. This is work that by Omar Den in my lab as a graduate student at the time. So we weren't thinking about, where it actually went. We were just thinking about, how about releasing stuff that's inside cells? Because people are saying, well, secretion, you have to engineer that, it's hard. We were thinking more like products, keeping cells in an optimal growth state. They could produce products better. That's how we were thinking at the time when we started thinking about this idea. So basically, here's the original design. So if I write down kind of old equations that we were thinking about at that time, this is just some meat on the bones, the quorum sensing story I told at the beginning with some actual equations where you have the number of cells and the quorum sensing molecule. And so if you plotted Q versus N, so how do you do this? I know is a pointer going to work here? Of course not. But if you took that to steady state, dq dt equals 0, and then plotted Q versus N using the right-hand side of the equation, you would get your S curve, right? So that's that's kind of how you would operationally do that if you wanted to pull up MATLAB and just generate that curve. And so you can see what we were going for is now at threshold, the cells are going to lyse, right? Because the quorum sensing thing goes on, and at quorum, they lyse. Now, you know, there was some debate about doing this experiment, right? Because people said, well, if all the cells lyse and die, what's the point of that? But if you came through the noise community in gene networks and synthetic biology, you would have been a little bit skeptical of the idea that you could kill all the cells. So you would at least have been in the mindset of, well, let's at least try it. I think it's kind of funny because it depends on how you pose the question. If you set up the question for a room full of academics and you said, we're going to trigger lysis such that it kills all the cells, you know, a lot of people will say, oh, you'll never kill all the cells. But if you set up the question to say, well, we're going to set it up so that when it lyses, only a few cells survive, and then it'll cyclically just cycle, everybody in the room said, that's never going to work. All the cells will die, right? That's just kind of how academics are. You know, I see somebody laughing there. So but anyway, I think we know that in ourselves, right? We're just skeptical of ideas. That is, in my mind, one of our biggest weaknesses, skeptical of ideas. And it comes out in crazy levels with new postdocs, at least that's been my experience. But you know, once they really try things, lots of things can happen in biology. You don't really know how anything works very well. Anyway, here's what happened. So we put this into E. coli. This is them growing up. Boom, they hit a quorum. A few cells survive. They grow back. They lyse again, and on and on. On and on until you get a mutation, depending on what kind of selective marker, Hoxoc stabilization you're using, whatever you're using. You can extend this with plain old antibiotics. I think it lasts about 4 or 5 days in microfluidic chips. Now we have low number of cells here. That helps you in terms of the evolution. But you know, after a while, they'll turn into pancake-shaped cells. We call them pancakes. I think there's a little bit of literature on that that evolution. But they turn into pancakes, but they keep doing this even though they're pancakes. So they're kind of round cells after 5 days. They change their morphology. Not surprising, you're lysing them. But they can't seem to get out from under the lysis circuit very easily. So they turn into pancakes first, and then finally the pancakes break the lysis circuit and then just exponential growth. But anyway, 5 days or something. We weren't even thinking about that at this time. So here's what the equations look like if you come from a quantitative background. It's just what I talked about before. There's the quorum sensing positive feedback system as a function of the number of cells. And the number of cells just is proportional to the magnitude of the quorum sensing, the AHL production. And then we have dNdt is exponential growth of cells. But then we just put a reset after lysis, right? So when it lyses, the number of cells must just go down, back down. So the circuit basically is going around this little circle in the lower right. So it's lysing, then going back down, lysing, and going back down. And so we just put a reset in the N equation to get you back over to there. And this poor man's model of that complicated thing is incredibly useful because conceptually you can understand what's happening. So usually I get a question about lysis circuit, thresholds, this or that. You know, I shouldn't say this because I'll get stumped today, but you know, usually I could kind of figure out what the answer to that question is because I have pretty basic conceptual understanding of how the dynamics work. And this is held up through a lot of different circuits we've built. For example, here's a hardcore analysis. Philip Bitton was a postdoc in my group, worked on just mapped out, you know, the onset of oscillations and in parameter space. Where do you expect oscillations? So the thing I like about this circuit, it's like our original oscillator, the intracellular oscillator that wasn't synchronized. It's very robust. It just seems to fire away in all circumstances. So, you know, we looked at it at different length scales. We've looked at multi-strain ecologies. I'll talk about that. And then I'll talk about in a mouse, it actually grows up, fires, grows back up. So it seems to be very, very robust. It works in chemostats. You know, you can use it to study evolution. It's just a cool circuit for a lot of different things. Oh, this is just kind of maybe driving home that point. Some things we've looked at using the synchronized lysis circuit in my group. The evolution one's cool over there at the left. That's in like a batch turbidostat, right? And you see it fires for a while, but then there's a mutation event, and you can go in and grab the cells and then sequence and find out what happened. How's it beating the lysis? And then we thought about trying to use that. We haven't had a whole lot of progress to build genetic circuits, because maybe you could come up with design criteria that would only be fulfilled if the lysis circuit beats, right? So maybe you could use this evolution process to build genetic circuits in a certain way, although, you know, we haven't had much progress on that lately, although the pandemic kind of shut things down. Okay, so the circuit ultimately ended up, you know, so we thought we would just publish the circuit like we have, but one of my students, Tal Donino, the guy in the middle there, who's speaking tomorrow in the same meeting, was doing a postdoc with Sangeeta Bhatia, and they were looking at bacteria as a delivery mechanism for solid tumors. And so at the same time, we had come up with the synchronized lysis circuit. Tal and Sangeeta were showing all this cool stuff about how you could orally deliver probiotic bacteria, and they would land inside metastatic liver tumors. And this is sort of right before all of the huge rage of microbiome, tumor microbiome papers. I don't know if you followed any of that, but, you know, it turns out that solid tumors are full of bacteria. And now it's gotten to a point, it used to be where I would say that people would say, well, where are the citations? Now I'm waiting for a paper that says this particular solid tumor doesn't have a microbiome, you know, because eventually there's going to be a exception to the rule. But now it's a sort of a very general phenomenon. Bacteria are finding their way into tumors, they're growing there. And so Tal and Sangeeta were working on this kind of right in the midst of a lot of this information about the tumor microbiome coming up. And so we got together and we thought, well, let's use the synchronized lysis circuit to deliver drugs in your tumor models. So here's just for the layman, and, you know, trust me, I'm not that far away from you in terms of what I understand about tumors. Maybe I've got some tumor experts on here, I don't know. But generally, what happens is bacteria, small numbers, circulate in the bloodstream. As tumors grow, they lay down a vasculature, and then the bloodstream feeds in, and bacteria enter tumors where they're protected from the immune system by the tumor. So it's not that bacteria are finding tumors on purpose. So you wouldn't use the word honing. You would use the word selectively growing, escaping the immune system. So it's just a safe haven. And, you know, Tal has some cool results where he shows once tumors are eradicated, the bacteria are cleaned up. You know, so you take their home away. It's like these tumors are a home, right? And so this little movie, my artist daughter helped me with this and made this little movie. And my other artist daughter helped me with that synchronized lysis. So it's good to have artists in your family. So this just shows the immune cells killing the bacteria if they don't enter the tumor. And growing up within the tumor. And there's some cool data out there now, and you can look around at other organs, you don't find the bacteria, you find them only in the tumor. So, people are still trying to figure out exactly how this is working, what's mediating, how is it working the best, what's optimal. Those kinds of details are still an active area, as you might imagine, since, only 7 or 8 years ago, when I started kind of talking about this, well, 5 years ago, I started talking in earnest about it, people would push back from the medical community and say tumors are sterile. How things have changed in 5 years. Nobody says tumors are sterile anymore. So anyway, what's cool about the bacteria is they can synthesize many therapies. At the time, we decided to try 3 classes of therapies. This hemolysin is just like a torpedo that destroys mammalian cells, burrows into the membrane. There's this chemokine that summons the immune system, and then apoptotic peptide, so a small molecule that will enter mammalian cells and then trigger apoptosis from the interior of the mammalian cells. So what's cool about the lysis paradigm is you don't have to engineer secretion mechanism for every single therapy you want to generate. It's just general. And you get what's called adjuvant effect, where the bacterial lysate also lights up the immune system and tells the immune system that there's something weird about this tumor, right? So the big problem with tumors, right, a big problem is that the body identifies them as self, right? So the immune system doesn't naturally attack them because it thinks it's you, right? So if you start releasing a bunch of bacterial lysate around a tumor, the immune system at least gets curious. What the hell's going on here around this big tumor with bacterial lysate? So anyway, we first in my lab tested with the hemolysin in the context of HeLa cells. As you could probably tell by now, I like movies. So I wanted to do a co-culture with HeLa cells and just see it working in action. So we put the hemolysin in to the synchronized lysis circuit and built a microfluidic device where we could co-culture HeLa cells and the bacteria in the same field of view. And then when we ran this experiment, you see the HeLa cells at the top, you see the bacteria at the bottom. When they lyse, you can see them releasing the hemolysin and killing the mammalian cancer cells, in this case, HeLa cells. So this is a nice proof of principle. It just visually shows exactly what's going on. It's not tumors, it's not a tissue model. One last thing we did before handing this therapeutic strain off to Tal was to do a batch culture experiment where we just mix the HeLas and the bacteria together. And we find that the viability of the HeLas only goes down when you have both the lysis circuit and the therapy. So this tells you that it's not just the bacterial lysing that's killing the HeLas. If the therapy can't get out via the lysis mechanism, it's not going to have any effect on the HeLas. You need to have both things going on. Okay, so this is the cool stuff that Tal and Sangeeta were working on, feeding bacteria to mice that have this metastatic disease, which is a very relevant, difficult model to cure. So it's not, there are different models in the space of animal cancer, and some are easier than others. This is a realistic model of metastatic disease, and that they localize to tumors. When we first tried it and compared with standard of care chemotherapy, we found that the bacteria didn't do much better than the chemotherapy, and the wind went out of the room. It was a little bit of like a bummer, right? So this is log scale. We'll look at some tumor stuff here. We're measuring right now, we're measuring the growth of the tumor as a function of time and days with a luminescence reporter that's related to the metabolic activity of the tumor. And so it's linear because you got exponential growth, right? So log linear scale here, exponential growth of the tumor. And so you see chemo's, slightly better than bacteria, but kind of the same. No. So, we're not the first to think about this, of course. Bert Vogelstein at Johns Hopkins has been talking about this for a while now. And we came across this paper at some point in 2001 in PNAS, where he talks about, the combination of bacterial therapies and chemotherapies being perhaps more useful than a monotherapy, because bacteria act from the inside out of a tumor, and chemotherapies typically have a problem diffusing into the inner interior of a solid tumor. So there's kind of a precedent idea here for why a combination might be the better way to go in terms of where they're acting within a tumor. So motivated by this, we tried combination. So here you see the combination that they delivered. They delivered the chemotherapy on days 0 and 21, right? And they delivered the bacteria on Monday, Wednesday, Friday. And even, I like to just joke, even the undergrads at MIT took the weekend off. So it's Monday, Wednesday, Friday, Monday, Wednesday, Friday, right? And so let's see what happens with the tumors when they did this. Combination therapy had a significant effect. So for around 16 days, we lowered tumor size. So I've looked at all this data myself. Since I'm a physicist, I'm kind of very leery of small numbers of things. And with mice, you have small numbers of things always. We've looked at the probability that the mouse dies. We've looked at the fraction of mice that die. We looked at everything. So I'm convinced with by this data. Very, very careful analysis here. So that dashed line in the middle is what the cancer community thinks is a significant reduction in the tumor size. So that's just decided by, the cancer people, the cancer pope or something. And so then for 16 days, we go below, the level where they say this is a significant response. So after a while, though, they return to growth. So after 16 days, the tumor begins to grow again. And actually, I don't have that slide in here, but what you will see is if you look at the tail of the green out there, it has the same exponent as chemotherapy alone, right? And it has the same exponent. So I'm looking at this green where it starts growing again in the middle, having the same slope as the red on the right. And what that means is likely we've lost the bacterial therapy due to some kind of evolution event or something, right? So it doesn't matter how many bacteria you add that haven't evolved every other day. If you've got bacteria in there that are growing exponentially, the lysing ones are never going to compete with the exponentially growing ones. So we plated some bacteria, we looked, and found loss of function after a while. This was still cool, still knocked down tumors for 16 days when combined with standard, so this 5-FU standard of care chemotherapy. And if you publish any paper in this space, you have to have 2 independent measures of a therapeutic effect. And so here was the second one. When you look at the survival curves of the mice, we were able to extend the lifetime of the mice by 50% for this realistic model from metastatic. So we're talking about stage 4 cancer here, where there's just my dad has it right now. There's very little that anybody can do, unless you're in a very, very tiny subset of cancers. Very, very little. Even the hot immunotherapies don't work very well right now for this. So, even pushing the needle a little bit with a probiotic bacteria that people might be able to orally take might be useful. There's a lot of mites. You start working in this space, you begin quickly to realize all the things that have failed. And it's very, 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 very difficult to get too excited about your little animal results. So I just want to sort of put an asterisk here that you have to stay grounded. I will say, as we backed off, you know, after we published this paper, here are some reviews, 2018, 2019. These are in Nature Reviews Cancer. And here's a synthetic biology approach that just appears prominently in those reviews. One is in the context of tumor targeting bacteria, all the different methods of targeting with bacteria. And another is in the context of all the different living therapies. The other big ones right now being oncolytic viruses. So people are designing viruses to deliver therapies. That's a hot area. And then CAR T, of course, that's the other big one. I put this slide up not to show off or anything, but really this is what the point I want to make is, of course, none of this was planned when we started playing around with circuits. And here we are in journals that I never thought I'd read. Nature Reviews Cancer, right? And here we are prominently, these are figures in the journals, right? About the work, two different versions of our circuit here. So I hope that this makes the point of the importance of, you know, again, I'm preaching to the choir, which is never dangerous, but the importance of the basic science and sort of just understanding and not, you know, always plan it out all the way to the end, you know. So anyway, maybe that's a better talk for the NIH or something. If you want to look at a really cool paper, so we were up against the synchronized, the previous study, we needed to do the co-therapy with chemotherapy, combined with chemotherapy. Tal has been working in his lab. He has a Nature Medicine paper. He also has a Science Translational Medicine paper on releasing immunotherapeutics so that you don't need the chemotherapy. So immunotherapeutics, let me just, my daughter made this movie. So basically the idea here is, okay, they fire and then the drug turns off this don't eat me signal so that the immune system attacks the cells. So basically cancer cells have a don't eat me signal, like all mammalian cells, right? And then, so the therapy in this case masks the don't eat me signal, so the immune system attacks the cancer cells. Really cool paper, use of nanobodies, which are a smaller antibody. So a portion of an antibody that's produced from llamas and alpacas turns out to be, you know, if you're following COVID, people are working with nanobodies in that space too. It turns out sharks have these weird antibodies too. But why those things? Because bacteria can express them. So bacteria, if you want to make antibodies, monoclonal antibodies, you got to use mammalian cells. Bacteria can make nanobodies. And this is just a piece of a nanobody that covers the don't eat me signal. Anyway, extremely cool study, not from my lab, from Tal. So if you want to just sort of look at the progression of this even further, you could check out that paper. So what we've worked on since that is genetic stability, given that we lose stability after a time. Of course, a lot of people are working on this in different ways. You know, I kind of have to stick with what I know best, and that is to perhaps engineer ecologies that can increase the stability, not necessarily the genetic stability, but the therapeutic stability. So what you really care about is how long can you deliver the therapy before, you know, evolution kills the whole enterprise. You don't really care if a bug evolves as long as another bug comes in and keeps delivering the therapy. That's the thinking. So go around Darwin as opposed to trying to beat Darwin. Anyway, the first thing we had on this, we kind of got for free when we started playing with two orthogonal quorum sensing systems. Right, so this is the RPA system. This just shows the circuit diagram for the two. The Lux systems are the system we've used all along on the left. And when we put these strains in a big microfluidic chip, what we noticed, which was weird, was that, you know, you would expect one of these strains growing faster than the other, it would just take over, you know, but it didn't happen. In fact, things were very stable. And so then we started, oh, wait a minute. You start thinking about it, yeah, the lysis, it's more complicated than that, right? You could have a lysis event, which opens up a spatial niche for the other cells. So it's not clear how the growth rate is going to kick out other cells. So yeah, this is a quote from Tal, actually, when he was in my lab. "A month at the bench can save you a day in the library." I just think that's funny because it flies in the face of everything we always heard as scientists growing up. So we did some agent-based modeling to figure out, you know, how's this working? And it actually turns out to be very, very robust to high differences in the growth rate. And so we did experiments with this and could determine, so this gray region, for example, is the region where you can get coexistence of two strains, alpha 1, wait, the resolution, I apologize, but that's alpha 1 and alpha 2 on the middle plot axes. Those are the growth rates in log. And so you see this big, very big region of coexistence that's gray due to the lysis phenomenon. So you get it kind of for free just by putting orthogonal systems in. And so just noticing that and then chasing it with some modeling, we went after this and showed it experimentally with two strains. Yeah, I'm kind of at 9:20. I better hurry up. When am I supposed to stop?
Perhaps let's wrap up in 2 minutes or something to have some time for questions right after.
Okay, I'll wrap it up. So let me just get to let me just say a few things about the last bit. I think I pontificated too much at the beginning. I apologize. But anyway, what we noticed was one of the systems was not really doing succinct lysing, you know, in a threshold manner, but you were getting this trickle behavior. Look at green. Green doesn't do kind of this punctuated. It was doing kind of this lysing, you know, I'm doing this with my hands. It was doing this kind of lysing. And so we followed up on that to figure it out. It turns out there's an inducer that can change the threshold for lysis. So what's going on here is just the threshold for lysis is way too low. And so you can change the threshold for lysis, you can control it. That's what this paper is all about. And then you can build kill circuits. You can build other kinds of circuits with this inducible synchronized lysis circuit. So this was published around the time the pandemic was hitting. So I don't think anybody read it, but it was a cool paper showing that you could induce this lysis circuit in a way that could generate kill circuits or different kinds of co-culture behaviors. And then rock, paper, scissors is an extension of this where we use collisins to kill previous strains. And the idea there is if you deliver on Tuesday you deliver green after delivering blue on Monday, green will wipe out blue, and you can just, through delivery on separate days, you could extend the lifetime of the therapeutic effect because the bacteria are killing the previous bacteria. And not only that, when you put all three of these together, you can get cyclical expression of 3 different therapeutics. So there are cases where the immune system can get primed by certain therapies, and then another therapy can be used to bring on the immune system once the first one is there. So you can deliver things in sequence using, say, the RPA system. Then that could be a useful way to deliver things. So you get interesting dynamics when you put these things together. So I'm just going to finish here. So it's just showing the sequential dosing. This is showing that we can increase the strain stability. It's all in the paper. I think you can understand what I'm talking about. And then this is just showing cyclical delivery when you put them all together. Now, we haven't tested this in any animal models, so we're looking forward and working with some people to test this in animal models. So here's where I'll wrap up. So I will say, Lev Simmering's been my collaborator since I joined. Well, 2 years after I joined UCSD, I've been working with him since 2003. Ruth Williams has worked with us quite a bit on some of the queuing system work that I didn't talk about here. And then Sangeeta, of course, is the person we began to work with on the cancer application. And now, you know, we talked to Tal, who's now a professor at Columbia. And then this is the end of the talk slide. Oh, look, I have a movie there. That's weird. Okay. So, okay, I forgot that I did that. But anyway, I guess I used to have this at the beginning when we would just all be standing around waiting to introduce the speaker. And I thought, boy, this is just a waste of time. Everybody's just sitting there, you know, so I would just have this movie of some stuff going on, and I would have a bunch of pictures of funny things going on in my lab if I could find them. But anyway, I'll conclude.
So thank you very much, Jeff, for the wonderful talk. Yeah, it was very interesting.
Hi, Jeff. Thank you so much for the talk. That was great. I have seen you talk before, and I'm always happy to see the work progressing. I was going to just ask, so all of your work so far depends on just this quorum sensing, picking up density. If you could communicate more than just, you know, the density of my population, my current quorum, what would be useful to communicate, and what could you do with that extra information?
Right. Outside of communicating the number of cells. So I think, you know, this is not a good answer to the question because I'm not going to go off the quorum sensing directly, but I'm going to say that doing some fractional quorum sensing, inverting the quorum sensing signal, doing those kinds of things is more in my immediate thinking, just trying to do things when it's just the opposite. So I need to stop this because it's causing feedback. Inverting the quorum sensing signal, for example, we've been thinking about ways in which cells do things when there's fewer cells. So having the threshold turned off. Fractional quorum sensing, I would say. But again, I'm not answering your question because I don't have a good answer. That's a very, very good question. And frankly, I haven't thought a whole lot about it.
Then maybe I could ask a quick follow-up, which is, so in, again, the earlier work, you're using this kind of intracellular toxin to lyse your cells. And in the rock, paper, scissors, you start using these intercellular toxins, these bacteriocins. Do you think that the intercellular toxins can in some way, I guess, replace the quorum sensing, in that you have a density dependent effect in some way from the fact that you've got a quorum of molecules producing these bacteriocins?
Ah, you're helping me answer your first question. That's great. So in fact, the rock, paper, scissors platform is independent of using quorum sensing or the synchronized lysis circuit. The colonies simply kill each other based on the number of cells producing the colicin. So yeah, indeed, this is an alternative to quorum sensing, is this bacterial warfare at the small colony level. So the rock, paper, scissors thing, you could drop any other kind of circuit in and still have it cycle between different strains that are doing different things. So it was sort of laid out there as a very general mechanism, as opposed to just relying on only quorum sensing and the synchronized lysis circuit. So you're absolutely right. It is a general platform that can be used for synthetic biology to cycle between different synthetic circuits that are in the 3 different strains, for example. Thank you for helping me with your first question.
Thank you so much for the question.
From Manish, yeah?
Yeah, sorry. Maybe I should have asked it directly because I can unmute myself. Just to ask, do you expect the lysis behavior to be different if the QS molecule was not freely diffusible. So if we're looking, I mean, a lot of QS molecules, for example, gram-positive bacteria, are peptides that need to mature as they, exit the cell, and it's only outside the cell that they mature and become and can act as quorum sensing molecules. So do you think this?
Yeah, that's a great point. So, since I started talking about this quorum sensing, people have begun to suspect that it's not as freely diffusing in and out, even in the classic AHL system, as maybe they thought originally. So I suspect that what we're seeing when we look at the waves of synchronization, for example, with quorum sensing, is something that's more like trigger waves because of the positive feedback. If you followed Jim Farrell's work, he highlighted this notion of trigger waves. Gurul-Suel has also followed up with some work on trigger waves, but basically a positive feedback flips over and then neighboring cells, more like dominoes than free diffusion. So I think it'll change the timescales, but it may even be that what you're talking about is already the underlying mechanism of what's going on in these AHL systems. I just haven't kept up with that as closely as I should have. People thought at the time I started working on it that it was free diffusion, but Manish, do you know if the classic AHL system, is it in the category that you're talking about?
Yeah, and from the description of the classic AHL systems, yes, it seems like many papers have described this as freely diffusible molecules. But what I was, in your understanding, where is the regulation coming from then in the AHL system? Is it the fact that there is some active export mechanism involved, or does the molecule actually get modified? Because that's where my original question came from. So.
Yeah, I don't think there's any modification, but I thought the question was more about active, so free diffusion or active transport. Is that what you're getting at?
No, I was getting at more. So, I was sort of thinking about a comparison with gram-positive peptide-based quorum sensing, where a peptide that's produced inside the cell will not activate the quorum sensing system within the cell. It has to go out of the cell, be treated by the proteases on the surface, and then it must re-enter the cell in order to behave like a quorum molecule.
So that sounds a little bit closer to our p-chimeric acid understanding the RPA system, where you do need an inducer. We called it we just thought of it as an inducer. And I sort of brushed through that really quickly because I was running out of time. But what we found, if this is the kind of system you're talking about, is that without p-chimeric acid, you don't really, the quorum is like one cell. So it's this absurd limit of the threshold being very, very low. But as you add p-chimeric acid, you could titrate the threshold for behavior. And so then you can play games with the dynamics of the quorum sensing system because you need this intracellular inducer, p-chimeric acid, which, by the way, comes in tomatoes and grapes. Interesting possibilities there too. I hope that helps a little bit.
Yeah, thank you.